I want you to visualize the very beginning of human life. You picture a developing embryo, right?
Right, yeah, that's usually the first image that comes to mind.
Exactly. And wrapped around it is this, the amniotic sac. You know, if you've ever watched a movie with a pregnant character, you definitely know about the water breaking.
Oh, absolutely. The classic dramatic movie moment.
Yeah. But we usually just think of the amnion as like a passive balloon. You know, just a simple bag of water that keeps the baby safe from bumps and bruises.
Right, a biological shock absorber.
Exactly. But today's deep dive completely shatters that assumption. I mean, it blew my mind. We're looking at this groundbreaking study published in Nature Cell Biology.
Yeah, by Wenke Hu and an amazing team of researchers.
Right. And they've basically mapped out a cellular metropolis. Like, it is not just a balloon.
It really is a metropolis.
Ugh.
What they did was they created a single-cell atlas of the human amnion during the first trimester of pregnancy.
Which is just a huge undertaking.
Oh, massive. And our mission today is to explore this dynamic, shape-shifting, communicating membrane.
Yeah.
Because, you know, once you understand the actual mechanics of how this tissue builds and rebuilds itself, you start to see the foundations of regenerative medicine.
Right. It's not just about pregnancy.
Exactly.
Yeah.
The mechanisms they uncovered here could eventually, well, change how we heal major physical trauma.
It's just wild to me how much complex biology is happening in what looks like a simple, clear membrane. So, okay, let's unpack this.
Let's do it.
Before we can understand the amnion's superpowers, we need to strip it down to its studs, right? We need the blueprint.
We do.
So the researchers looked at samples from what are called Carnegie stages 16, 17, 19, 22, and 23.
Which sounds very technical.
Yeah, what does that actually mean in terms of human time?
Well, Carnegie stages are basically a standardized way embryologists track physical development. It's based on physical features rather than strictly like days or weeks.
Because early development can vary slightly from person to person.
Exactly. But roughly speaking, the stages they looked at translate to between 5 and 9 weeks of pregnancy.
Okay, so very, very early on.
Super early.
And to figure out what was happening inside this membrane, the paper says they used a technique called single-cell RNA sequencing.
Yes, RNA-seq.
Right. And they isolated over 14,000 individual cells.
14,027 individual cells, to be exact.
Ooh.
And to understand why this technique is so revolutionary, imagine you have a massive, complex smoothie.
Okay, a smoothie. I'm with you.
Old technology would just blend it all up and you'd get a readout saying, you know, this smoothie is 20% strawberry, 50% banana, and 30% kale.
Right. Just a broad average of everything in the cup.
Precisely. But single-cell RNA sequencing allows you to pull out every single piece of fruit, identify it, and read its individual active instruction manual.
Oh, wow. Because every cell in your body has the exact same DNA, like the same master hard drive.
Exactly.
But they aren't all reading the same files at the same time.
You nailed it. RNA is the active copy of those files. By looking at the RNA, the researchers can see exactly what machinery the cell is actively building at that exact moment.
It tells you what its actual job is right then.
Right. It tells you a cell's identity.
Yeah.
And when they read those active instructions, they found that this simple, bag of water actually consists of 2 primary layers separated by a thick basement membrane.
Okay.
And it contains 6 major cell types.
Wait, 6? So it's definitely not just a sheet of plastic.
Not at all. Let's look at what is actually building this thing.
Yeah.
First, you have the amnion epithelial cells.
The AECs.
Right. These are the surface cells lining the cavity, actively making the amniotic fluid.
Okay, the inner layer.
Then second, you have the amnion mesenchymal cells. These are embedded deeper in the matrix and act as like the structural scaffold.
Got it.
Third, you have fibroblasts. They provide even more heavy-duty structural support by laying down collagen.
Okay, so that's 3. What are the others?
Fourth, you have macrophages, which are immune cells.
Oh, interesting.
And finally, the 5th and 6th types are actively dividing stem cells. So both mesenchymal and epithelial stem cells.
Which are just rapidly multiplying to provide raw material as the tissue grows, I assume.
Yes, exactly. They're the factories.
Okay. To give this a physical shape in our minds, let's think of the amniotic sac like a highly advanced self-building swimming pool.
Oh, I like that analogy.
Yeah. So the epithelial cells are the flat, tightly connected, waterproof tiles on the inside, right? Actively pumping the water into the pool.
Spot on.
And then the mesenchymal cells and fibroblasts are the concrete and the rebar in the walls holding the entire shape together.
Yeah, the heavy lifters.
And the stem cells are the factory churning out new tiles and new rebar. But wait, I need to stop here for a second.
Sure. What's up?
You mentioned macrophages. Aren't those immune cells? Like the Pac-Man cells that go around eating bacteria?
That is their most famous role, yes.
So at 5 weeks of pregnancy, in a sterile womb, what on earth are they fighting? Are there that many infections happening that early? Like, why does a water balloon need a dedicated security team?
You'd completely think it's just about infection. But what the researchers found is that in fetal development, their primary role isn't fighting off invaders at all.
Really? What is it then?
It is tissue remodeling.
Remodeling, like a renovation crew.
Exactly like a renovation crew. The amnion is expanding rapidly to accommodate a growing embryo.
Because the baby is getting bigger every day.
Right. And you can't just stretch concrete and rebar endlessly.
Yeah.
You have to tear parts of it down and rebuild it bigger.
Oh, wow.
So the macrophages are the demolition crew. They move through the tissue, secreting enzymes that dissolve old matrix, clearing away dead cells.
Just literally eating up the old walls.
Yeah. And releasing chemical signals that help build new blood vessels and structural scaffolds. The tissue isn't just a passive barrier. It's an active construction site.
That completely flips how I view an immune cell. I mean, it's less of a security guard and more of a structural engineer. Okay, so we have our blueprint. We've got our waterproof tiles, our structural rebar, our actively dividing stem cells, and our demolition crew macrophages.
A very busy pool.
Extremely. But if we have this relatively rigid pool made of tightly connected tiles and concrete, how does it physically stretch? By week 9, that embryo is significantly larger.
Oh, massively larger.
So how does the sac not just shatter under the pressure?
Well, that brings us to the most dynamic part of the study. The cells don't just stay in their assigned roles. The building literally shapeshifts.
The paper calls this pseudo-time analysis, right?
Yes.
But how do you track a cell shapeshifting over weeks if you're just taking a snapshot of a tissue sample at one specific moment in time?
It requires some incredible computational biology. Because, like you said, you can't watch a single cell change over weeks in a static snapshot.
It's just frozen in time.
Right. So researchers use algorithms like RNA velocity. Think of it like taking a high-speed photograph of a busy highway at night. You can't see the cars actually moving in a still photo, but by looking at the blur of the headlights or measuring the fresh exhaust plumes coming out of the tailpipes, you can calculate exactly which direction a car is heading and how fast.
So the exhaust plume in a cell is like the freshly made RNA versus the older RNA.
Exactly. By comparing newly transcribed RNA to mature RNA, the algorithm predicts where a cell came from and what it's actively turning into.
That is so smart.
It's brilliant. And what they found in that cellular blur were intermediate state cells. They found cells that were reading both the epithelial instruction manual and the mesenchymal instruction manual at the exact same time.
Wait, meaning the waterproof tiles are turning into concrete rebar?
Yes.
That is insane.
It is a biological process called epithelial to mesenchymal transition, or EMT. And they found the reverse happening too.
Swap back and forth.
Mechanically, what happens is those tightly packed, flat surface tiles literally dissolve the biological glue holding them to their neighbor.
They unstick themselves.
Yeah, they change their physical shape into these elongated crawling structures, and they migrate down into the deeper concrete layer to become structural rebar.
That is wild. So the amnion undergoes this constant dynamic remodeling because it has to grow massively without rupturing.
Exactly.
If the wall is getting thin, the surface tiles just unstick and dive down to reinforce the foundation.
Precisely. They also found the surface cells turning into the demolition crew and epithelial to immune transition.
Okay, but here is the finding that made me do a double take when I was reading the prep.
Oh, I bet I know which one.
They found a specific group of cells that were expressing ectoderm markers, specifically a gene called SOX2.
Yep, the amnion ecta cells.
Why is that a big deal? For the listener, what does SOX2 do?
Because SOX2 is a master switch for creating neural tissue. It's a gene that tells a cell, hey, we need to start building a brain or spinal cord or nerves.
Wait, so why is a protective water balloon trying to grow a brain?
It's not trying to grow a brain, necessarily, but it's a fascinating look into pluripotency. You know, the raw, uncommitted potential of early life.
The ability to become anything.
Right. Remember, the human amnion originates from the exact same cluster of early cells, the epiblast, that gives rise to the entire embryo.
Right. They share a common ancestor, basically.
Yes. So finding these neural markers lighting up in an amniotic sac shows us that these tissues retain a shadow of that incredible plasticity. They haven't completely forgotten their origin.
They still have the potential to be almost anything. I love that.
It's amazing.
But I want to go back to that physical shapeshifting for a second, the EMT, where the tiles turn into rebar.
Okay, yeah.
The paper mentions that this process triggers localized inflammation. Now, usually when we hear the word inflammation, we think it's a bad thing.
Right, like an injury.
Yeah, we take anti-inflammatory drugs. Doesn't inflammation damage a fragile early pregnancy?
Well, we've been conditioned to think of all inflammation as a disease state, but mechanically controlled, localized inflammation is a vital tool.
Really?
Think about how rigid structural tissue is. If you just pull on it, it snaps. But inflammation brings a rush of fluid and specific enzymes to a targeted area. It literally softens the tissue matrix.
Oh, I see.
It turns cold, hard plastic into warm taffy.
So the cells are intentionally causing a microfire to soften the wall so it can stretch and expand without tearing as the fetus kicks and grows.
Precisely. They are temporarily destabilizing their own architecture to allow for safe expansion.
Okay, let me make sure I have this straight, because this is a lot.
It is a lot.
We've got a waterproof pool that is constantly tearing itself down, softening its own walls with inflammation, and having its surface tiles detach and migrate to the basement to become structural support.
It sounds chaotic, right?
It sounds like absolute chaos. How does the left hand know what the right hand is doing? If too many tiles detach at once, the sac leaks.
Right.
If the inflammation goes too far, the tissue fails.
You've hit on the exact reason why intercellular communication is so vital here. For these shape-shifting transitions to happen in perfect harmony, the cells need a highly coordinated communication system.
A group chat.
Exactly, the cellular group chat. The researchers meticulously mapped out these pathways. They looked at which cells were producing specific signaling molecules, think of them as chemical keys, and which cells had the matching receptors or locks to catch those signals.
And one of the most critical communication networks they uncovered involves bone morphogenetic protein, or BMP signaling.
Yes, BMP4 specifically.
It's crucial for triggering all this growth and differentiation. But here's where it gets totally counterintuitive for me.
Let's hear it.
I would naturally assume the epithelial cells, the ones right on the surface, touching the amniotic fluid, interacting with the environment of the growing baby, are the ones dictating the growth, like they are the front line.
It makes complete logical sense to think the surface runs the show, but the data showed the exact opposite.
Right. The study found that the mesenchymal cells, the structural guys buried down in the basement, are the ones manufacturing and sending the primary BMP4 signals.
Yep.
And the surface epithelial cells are primarily receiving them. The basement is telling the roof what to do.
Yes. And it gets even more distributed than that.
Oh, boy.
If you look at another major pathway, the TGF-β signaling, the paper shows that the macrophages, the demolition and renovation crew, are acting as the directors there.
The immune cells are directing it.
Yeah. They are sending TGF-β signals down to the mesenchymal cells to coordinate their growth.
Okay, so the demolition crew is commanding the structural rebar, and the structural rebar is commanding the surface tiles. Why is the communication network built upside down like that?
It all comes down to mechanical tension.
Tension.
It makes perfect mechanical sense when you realize what these deeper cells are actually experiencing. The mesenchymal cells and the macrophages are embedded deep within the structural matrix.
Okay, they're inside the walls.
As the embryo grows and the amniotic fluid volume increases, the sac stretches. The surface cells might just feel fluid, but the deep structural cells physically feel the tension. They're literally being pulled apart.
Oh, wow. They are acting as mechanical strain gauges.
Exactly. They translate that physical stretching into a chemical signal.
That makes so much sense.
When the rebar feels too much tension, it secretes BMP4 and TGF-β, which diffuse up to the surface and tell the epithelial cells, hey, the structural integrity down here is reaching its limit. We need you to multiply, or we need you to undergo that shape-shifting EMP process and migrate down here to reinforce the wall before we tear.
That is brilliant engineering. I mean, the leadership and direction aren't coming from the top. They are distributed based on who has the most critical data.
Exactly.
In this case, the cells feeling the physical stress are the ones pulling the alarm.
It is a perfectly coordinated feedback loop driven entirely by physics and translated into biology.
Okay, so we've mapped the blueprint, we understand the incredible shape-shifting plasticity, and we've decoded how mechanical tension drives their communication. This is beautiful biology.
It really is.
But how do we actually harness this? Like, the paper talks about taking this knowledge and applying it outside the womb into the lab.
Yes, the researchers didn't just map the human amnion, they compared it.
Compared it to what?
They looked at single-cell data from non-human primates. specifically cynomolgus monkeys,
Yeah.
to see how evolutionarily conserved this is. But more importantly for medicine, they compare the human data to several different in vitro, lab-grown, stem cell-based models of the amnion.
So they're checking if the stuff we grow in a petri dish actually acts like the real thing.
Exactly. And the findings reveal a massive divergence based on the physical environment the cells are grown in.
The paper notes a major difference between growing cells in a 3D environment versus a 2D environment. What is the physical difference for the cells?
Well, a 2D culture is exactly what it sounds like. It's cells grown flat on the bottom of a hard plastic petri dish. A 3D biomimetic culture involves suspending the stem cells in a three-dimensional gel matrix, sometimes within microfluidic devices where fluid is actively flowing over them.
Giving them a real environment.
Right. And the study found that when scientists grew stem cells in those 3D environments, the cells functionally resembled very early human amnion stages, around Carnegie stage 7.
Okay, very young cells.
But when they grew them flat on a traditional 2D dish, the cells skipped ahead and reflected a much later stage of development, around Carnegie stages 11 through 16.
Wait, why does the flat dish make them act older?
Because it lacks the very thing we just talked about, physical tension and depth.
Ooh.
In a flat dish, there is no 3D matrix for the macrophages and mesenchymal cells to embed within. There is no fluid pressure stretching a membrane.
So they don't feel the pull.
Without that mechanical resistance, the feedback loop breaks. The cells don't get the signals to hold their early, highly plastic state, so they just rapidly mature.
So if a researcher's trying to figure out the root causes of early miscarriages, or, test a new drug to help embryos implant securely, and they do all their testing on a flat 2D petri dish.
They are essentially studying a teenager when they think they're looking at a toddler.
Wow.
The tissue is functionally at the wrong stage of its life cycle.
That feels like a massive breakthrough for researchers. It tells us that context dictates biology.
Absolutely.
You cannot recreate the regenerative superpowers of these cells if you don't first recreate the physical physics of their environment.
Careful selection of the model system is paramount. If you want to accurately reflect real-life formation, you must match the physical environment to the biological question you are asking.
Man, we have covered some truly incredible ground today.
We really have.
I mean, we started by looking at a simple, clear sack of water, the thing we just casually call the water breaking. And by reading the active instruction manuals of over 14,000 individual cells, we uncovered a complex, sensing, shape-shifting metropolis.
It's beautiful.
We saw rigid surface tiles unsticking and migrating to become structural foundation. We saw demolition crews orchestrating growth. We saw cells holding the hidden potential of neural tissue. And we saw how physical tension drives a brilliant bottom-up communication network.
It really is a marvel of cellular plasticity. And, that plasticity brings us to the ultimate why. Why mapping this tissue matters to you, the listener.
Right, the practical application.
The human amnion is famous in the medical world for having incredibly low immunogenicity.
Which means what, exactly?
It means it doesn't trigger immune rejection when transplanted. It also has potent anti-inflammatory and antimicrobial properties.
I know doctors already use leftover amniotic membrane tissue for wound healing, right? Like treating severe burns or even repairing ocular surface disorders. They literally put it on the eye to heal scars.
They do, and it's highly effective. But until now, we were essentially using a black box.
Like we knew it worked, but not how it worked?
Exactly. We knew the tissue worked, but we didn't fully understand the cellular mechanics of how. Thanks to Wenke Hu and this team, we now have the high-resolution single-cell atlas. We have the exact instruction manual.
So we aren't just limited to using donated tissue anymore.
Right. Imagine a future where we take this deep knowledge of the BMP4 and TGF-β pathways, this understanding of how mechanical tension triggers shape-shifting, and we apply it to adult cells.
Oh, wow.
Imagine instructing a patient's own cells to mimic the amnion's communication network. We could potentially program our own tissues to recreate that embryonic state of rapid, scarless healing to repair massive physical trauma.
We could teach adult joints or damaged organs how to renovate themselves, just like the amnion does.
Exactly.
We could instruct our bodies to build the pool all over again.
It's a game changer.
Well, thank you for joining us on this deep dive. The next time you hear about a baby being born or the water breaking, you'll know there's an entire, unseen, calculating metropolis in there that made it all possible. Keep questioning the hidden complexities of the world around you.